My name is Nathan Escher, and the scientist I've chosen is Dr. Andrew Pollard. So, Andrew Pollard is a British scientist who's helped in creating vaccines such as the Oxford AstraZeneca COVID-19 vaccine. I chose him because of his scientific achievements and also because he really values communication within his field and just making the science very easy for everyone to understand, which is something that I personally connect with. So, a little background about Andrew Pollard was that he was born in England in 1965, and growing up, he was always curious about how diseases spread and how our bodies fight back against them. He went on to work and study medicine at the University of Oxford and also spent some time teaching at the University of Bristol. He is currently the director of the Oxford Vaccine Group and a professor of pediatric infection and immunity at the University of Oxford. His field is immunology, and the main question he's interested in answering is why do some people feel really sick from a disease while others with the same disease barely show any symptoms? Outside of science, Andrew Pollard has a lot of interesting hobbies as well. He's an experienced mountain climber and has climbed mountains such as the Himalayas, and he was also the deputy leader of a British Everest medical expedition back in 1994. In his spare time, he's also written many books and over 300 scientific papers about vaccines for the general public. So the reason why I admire Andrew Pollard is because he has used his research on vaccines to solve many problems, especially for people who don't have access to very good healthcare. When the Oxford AstraZeneca vaccine was released, Andrew Pollard played a huge part in advocating that these vaccines would be sent to countries and places that couldn't really afford expensive vaccines. I feel a personal connection to that because when COVID-19 first came around, some of my own family members were struggling financially and getting the vaccine wasn't very easy for them. So seeing someone like him try to make the vaccines more available to everyone is something that I personally connect with. So here's a figure I chose from one of his most recent papers that was published in 2024. The paper was about understanding why some people get sick after being exposed to S. typhi, which is a bacteria that causes typhoid fever. The research question for this paper was, can we detect early immune differences between people who get sick and people who stay healthy? So the figure you see here was a human challenge study where some volunteers were given S. typhi. The experiment was focused on investigating monocytes, which are a kind of white blood cell, and looked at three different subtypes: classical, intermediate, and non-classical. They used flow cytometry, which is a technique where you label cells with fluorescent tags so they can use a machine to count all the different cell types. The control group in this experiment was people who didn't show any typhoid symptoms. So, to explain this figure, if you look here in panel A, we're shown three monocyte groups that were separated based on CD14 and CD16 markers. Panel B shows that before the typhoid infection, most people had classical monocytes while less people had intermediate and non-classical monocytes. Panel C shows the percentage of classical monocytes over time with both groups staying mostly stable. In panel D, the people who did get sick, which is shown by the red highlighted line, shows a little spike in intermediate monocytes, while the healthy group, which is shown by the blue highlighted line, stays stable throughout the graph. Panel E shows the percentage of non-classical monocytes over time, which again remain mostly stable for both groups. And finally, panel F shows that in the group that was diagnosed with typhoid, the percentage of intermediate monocytes increased while the classical and non-classical monocytes remained unchanged. The conclusion for these figures was that S. typhi triggered an increase in intermediate monocytes in symptomatic volunteers while the other monocyte types remained unchanged. Andrew Pollard's discoveries have led to the creation and improvement of vaccines for diseases like meningitis, typhoid, and COVID-19. His next steps are focused on making vaccines that don't require refrigeration so they can send more vaccines to low income areas. So, to sum up everything I just said, Andrew Pollard is an immunologist that does vaccine research at the University of Oxford. In his free time, he likes hiking and spending time outside, and he is a huge advocate for vaccines being accessible to everyone, especially for people in low income areas.